Okay, terminology is really important. I'm going to need you guys to work on spelling. I know it's hard. I know. So this is my thing. I have a rule for spelling. Now it could be slightly misspelled as long as it doesn't like refer to something else, right? So say for instance you're spelling out like stoma, and you write out stroma. Not the same thing. Not the same thing because stoma and stroma got two different meanings in biology, right? So that's why it's going to be very important to like get as close as you possibly can to spelling, and we're going to just go with that. If you get close enough to where I know what you're saying and you're not inferring something else, all right. So when it comes down to microorganisms, so there are some instances where we may say microorganisms, microbes, same thing. All right. So microorganisms are organisms that are too small to be seen with the unaided eye. So we need something to look at them with, right, like a microscope. And so we're talking about these microbes and include bacteria, which is going to be the gist of our course. Fungi, also in our course. Protozoans in the course as well. Some microscopic algae, absolutely in the course. Viruses, but we don't look at prions. Prions are cool, but we don't really have a lab for prions. We'll talk about them in a little bit, but for the most part, we don't have a lab for those. But all the other ones, we're going to do something with those. All right? Now, when talking about microbes, people think for the most part that they're bad, and that's not always the case. The reason why you think they're so bad is because there's only a handful that are problematic or pathogenic. Pathogenic means they cause diseases, right? For the most part, they're actually quite beneficial. It's like sometimes we have like a small section that's really, really loud, and it makes everything look bad. It's like, no, not really. It's just that it's that small group that's just pathogenic, and they make all the microbes look bad. So when talking about microbes, something called food spoilage. Yeah, if you leave your food out, you're supposed to put it away, keep it in refrigerated temperatures, right? And I'll teach you guys why that happens. The basis of food chain in aquatic environment. So in the ocean, right, we have microorganisms that exist like algae that are going to be very important when talking about photosynthesis, things like that, right? We also have microbes that decompose organic waste, which makes sense, right? Because fungi are decomposers. Yeah, fungi are decomposers that break down organic waste. And then some of these microbes can actually incorporate nitrogen gas in the air into organic compounds. That's really important. So nitrogen gas, periodic table right here. So it's smack in front of it though. N, right? N2 is nitrogen gas. Can you? Oh, it's locked. I'm not playing with it. But yeah, N on the periodic table, nitrogen gas. And then some of these microbes can generate or make oxygen through photosynthesis, like I mentioned with algae, right? Algae can undergo photosynthesis and photosynthetic microbes. All right? Now, we also have some microbes that can produce chemicals such as ethanol, acetone, and vitamins. And so ethanol, acetone is going to be something we talk about through fermentation. We learned about that before, right? Fermentation is pretty cool. Also, with the fermented foods, you include vinegar, cheese, yogurts, alcoholic beverages, and bread, right? And then last but not least, we have some like applications, some really good applications for microbes. So some can be used to produce products used in manufacturing, right? Such as cellulose. What's that? Cellulose. I heard that before, right? Cellulose is found in plants. Absolutely. So cellulose is the most abundant polysaccharide. We'll get that in chapter 2. But it's found in plants and it's the most abundant. You find it everywhere, right? There's a plant somewhere, right? There's a plant somewhere. Also, we use microbes for disease treatment, such as insulin production. All right, but here's some other cool things that which we can see microbes are going to be used for. So I really like this slide. This slide's pretty fun to me. I add this slide to the PowerPoint because it's something that's really interesting that we don't think about. So we have this fungus called trichoderma. So trichoderma is a fungus that's going to be used for fading the denim color of the jeans. All right, so it's actually used as a biological compound in a manufacturing plant to fade denim jeans. Real thing. You also have Muconocidobacter xylem. So Muconocidobacter xylem is a microbe that's going to involve related hops. How does it do that? Well, it's going to be actually able to make cellulose. It makes cellulose, and cellulose is made from polysaccharide found in plants. How does the plant? Make sense? Yep. And then you have mushroom peroxidase. Peroxidase, what does that sound like? Peroxidase? What's that sound like? Peroxide, right? Peroxide. Okay, so I'll give you like a little bonus right now. So peroxidase is an enzyme. Anytime something ends in ASC, I'm gonna say this all semester so that way we can know this because it's gonna be very important in coming chapters. Okay? Anytime you see something ending in ASE, it's an enzyme. Alright, so peroxidase is an enzyme, and then the names are kind of tell you what it's gonna be doing too. So peroxidase is an enzyme that's gonna break down peroxide. So that's why menstrual peroxidase is important with bleaching the denim jeans. Right? Have you guys ever used peroxide to bleach your hair? Yeah, don't do that. But you can do that, right? The reason why I say don't do that is because you don't want to really mess up your hair integrity, but like you can. Just don't. Alright, and so then Escherichia coli. E. coli is the model organism in microbiology. We know a lot about it. We know a whole lot about E. coli because we know its whole genome, all the genetics of it, right? We're learning a lot about its proteins too. But with E. coli, it has the ability that we can modify the genes and insert a pigment molecule to allow us to indigo. So here you have a bacterium. This is the E. coli bacterium. And you see that little blue pigment on the inside, the indigo pigment on the inside? That's because they added the genes to it to make it like look indigo. And like genes are indigo, right? That's how you can make the dye. You can make lots and lots of the dye. And the reason why that's cool is because E. coli makes a copy of itself every 20 minutes. So if I can make the dye every 20 minutes, naturally, wouldn't I want to do that? It doesn't really cost me anything. I can just keep making it over and over and over and over and over every 20 minutes because E. coli has that type of bacterial replication cycle. And then last but not least, bacterial polyhydroxyalkanoates. That's PHAs. Somebody got time to keep saying that. Polyhydroxyalkanoates. No. PHAs. Alright, bacterial PHAs. Those are going to be involved in the plastic on your zipper. Like I got a zipper on my pants, right? The little zipper part. Yeah, that's the bacterial PHAs is going to be responsible for making the plastic for that. Pretty cool stuff, right? The stuff that you wear every day. You're like, that's bacteria involved in my clothes? Absolutely. And it's totally fun, alright? Alright, so let's talk about the microbiome. So what even is that? So the microbiome basically is a group of microbes that live on or in your human bodies. Right? I tell people all the time, normal microbiome is different from person to person. But I will tell you though, what's really cool is that people within the same household, their microbiomes are similar. Which is cool. Alright, so an adult human is composed of 30 trillion body cells. We know body cells, right? Where do you get body cells from? There's a process that starts with an M that makes body cells. Mitosis, great job. So we get body cells from mitosis, right? So when talking about those body cells, we have 30 trillion of them. About 30 trillion of them. And we have about another 40 trillion bacterial cells, right? So I have my regular old body cells, and I have a lot of additional bacterial cells to go along with that. But those bacterial cells are cool though, they keep me healthy, right? So we're gonna do a lab in, I forgot it's like gonna be maybe around like April, where we're gonna be doing a swab of our noses, and we're gonna be looking at this on a staph plate. And so we're gonna see what type of staph we naturally carry, because everybody naturally carries Staphylococcus epidermidis. Staph epidermis, that's gonna be on your hands. That's cool, that's okay, right? But some of y'all carry Staphylococcus aureus naturally. Now Staph aureus, the MRSA one, not that one, not MRSA, but the Staphylococcus aureus normally. I found out last semester that I carry it naturally. It's all up in there. And I'm like, oh, but it won't make me sick though, because I carry it, right? I carry it, I'm good, it shouldn't make me sick. But let's say if I go to like a newborn baby, like, oh, look at the little baby, and I'm playing with the baby and I'm sick, and I'm like, if the microbe hits on the baby, that baby can get sick. That's why I tell people, do not kiss newborn babies, do not kiss small babies. Do not handle small babies, that's not yours. Alright, because you have your own little microbes, that baby doesn't have theirs yet. They only got what their mom gave them. Alright, so please be mindful of that. And so we're talking about these microbiomes, they help to maintain good health, right? They can prevent growth of pathogenic microbes, so Staph epi on my skin is good. Right? Because it can help prevent like other harmful things from actually growing on my skin. Right? So they're competing for the same resources and hopefully the Staph epi wins. And so then you have these microbes that may train your immune system to discriminate threats, which is good. You want your body to identify threats, right? You want to identify threats and to eliminate them. So when we talk about the normal microbiota, this one is going to be the collection, same thing, the collection of acquired microorganisms in or on a healthy human being. So you're probably gonna get that before birth, right? It begins to be acquired before birth because your mom's gonna pass on that immune system to you, right? The innate immunity that you get from your mom. They may colonize the body indefinitely, so you may carry it forever. Or you may have it like fleetingly, so that may be making them transient microbiota. So transient means like you may have it at certain times and sometimes you don't. Just depends upon where you're at. Right? Like let's say you change like climates, for instance. Let's say you move from one country to another. Well, if you come across this particular microbe in this one country and it's not in this other country, You may have it until some time passes and you may not have or carry that microbe any longer because of that climate change. Or you changing climate. So, colonization, what does that mean? Colonization means that it can only occur at body sites that provide nutrients and the right environmental conditions for the microbes to grow. Right? So some microbes need certain things. Right? Staph epi, I keep throwing that one out because staph epi naturally grows at human body temp. I'm a human and it's on my body, so that makes perfect sense, right? E. coli grows also at human body temp, and E. coli hangs out in the gut, which makes sense. I'm a human body temp, E. coli in my gut. Great, all right? So now let's talk about how do we name and classify these microbes? Are we good? Are we feeling pretty good so far? I'm going too fast, slow me down, okay? One thing I will say too, I'm about to slow myself down only because I want to make sure that as you guys are writing, I have to still think. That if I can see that you're still writing, that means you're still thinking. I don't want to keep talking while you're still thinking, right? That can make thoughts kind of mesh together. I do that too. You talking about one thing, like I ear hustle. So like if I'm doing like the real thing I'm supposed to be doing, and then I'm listening to this conversation too, I mess around and start writing both things together. Like that's why I shouldn't be ear hustling, all right? So that I can focus on one thing. So I'm gonna pay attention to make sure that I'm not continuously talking. I'm just kind of like finish that in one thought. Alright? Alright, so we're gonna talk about the nomenclature, scientific nomenclature. You guys heard that word before? Are those words before? Scientific nomenclature? You have, right? And then we're gonna talk about how they use a two-part naming system. The genus name and the species name or the specific epithet. Back when I was in school, it was species. Now it's specific epithet, which is the same thing. Well, slightly different, but I'll tell you why. And then we're gonna talk about the major characteristics of these groups of microorganisms. And then three domains. We know what domains are. Y'all remember what are the domains of life? Not yet? Okay. We'll get it. We'll get it. Alright, so Carl Linnaeus or Carolus Linnaeus, we've heard of him before, right? No. Oh. Alright, so Carolus Linnaeus, he's the father of taxonomy. Alright, so Carolus Linnaeus is the father of taxonomy. He established the system of scientific nomenclature in 1735. Alright, I wasn't thought of, you wasn't thought of, your parents weren't thought of, mine wasn't either. Alright, 1735. So, the work that he did that was really, really important in the way that we name microbes, right? He was the person that came up with the idea that organisms should have two names. And that's going to be their genus name and their specific epithet. Alright, so let's see some examples of this. The genus name and a specific epithet. Alright, so when talking about the scientific names, a few tips first. They must always be italicized or underlined. Okay? Can you write in italics? Like, you can write. I can't. I've been trying. I try my hardest to slant it, but I just cannot italicize my handwriting because you can't. Alright, so on the computer, you're italicizing on your computer. When you handwrite it, you underline it. Right? What's the name for humans? Scientific name for humans.
Homo sapien.
Thank you. I'm like, I know, come on now. I know you know something. We got it. Homo sapiens. Is this okay? Can I write this like that?
No, underline.
No. So I got the part right. So the first letter of the genus name should be capitalized. So I did that right. That's good. The species or the specific epithet's first letter should be lowercase. I did that right. But what do I need?
Underline.
Underline it. Alright. So what about this one? Canis lupus familiaris. No, is that a I? Hold up. Alright. Canis lupus familiaris. Is that okay to write this like that? Like you got the capital C, capital L, lowercase f. Can I write that?
Underline.
Yeah, I do need to underline, but like, can I just write it like that though?
No.
Nah, that still needs to be lowercase. Alright, the reason why I put this out there is because you will see some things like this, but I will not ever test you on that one, only because I why. Right, we only see this because Canis lupus familiaris is the scientific name for dogs. Right, dogs and wolves have the same scientific name, Canis lupus. It's just that dogs are domesticated, so they put familiaris because it's domesticated dogs. Alright, so just to throw that out there, so if you see it, you won't be confused. So all it is the genus name, the species name, and the subspecies. Or genus, specific epithet, subspecies. Alright? But for us, we don't care. We do Canis lupus, we do Staphylococcus epidermidis, we do Pseudomonas fluorescens, right? All those different microbes. Alright, so another thing that I wanted to throw out is that scientific names are Latinized. Do you guys know why they're Latinized? Have you ever heard this, that Latin is a dead language? What does that mean? What's your name?
People are learning. Oh,
What's your name?
Asia.
Say it again?
Asia.
Zaisha? How do you spell that? That's pretty.
Asia with a B in the front, like B-A-S-I-A.
Nice. Nice. Okay.
Either less people are learning it or speaking it. Or
Okay.
it's being forgotten.
So that's a good point, but that's not what that means though. So let's talk, cause less people actually are speaking Latin, which is unfortunate because you really wanna learn Latin. If you understand Latin, I promise you, biology is a breeze. I learned that late. I wish I would've learned that when I was younger. I learned it now, but like, it's like, dang, I should've really learned it in college. But when talking about Latin, Latin is a dead language. It means the same thing everywhere, right? So if I say this Latin term in America, it's gonna have the same meaning in Asia. If I say this Latin word here in Asia, it's gonna have the same meaning in Ghana. If I say it in Ghana, it's gonna have the same meaning in Australia. If I say it in Australia, it have the same meaning in Canada. So it's like no matter where I'm at, it's gonna have the same meaning. Alright, now the reason why I say that if you learn Latin, like if I had a school, I would make sure that my kids learn Latin because if you understand that, you'll understand science. You'll understand the world, period, because most of the English words are Latin based anyway. Alright, you'll be able to move between languages if you understand Latin. Alright, but the last thing is that most names in scientific naming are gonna be for the description of it or under science. Let's take a look at that. So Escherichia coli, E. coli, Escherichia. Big word. That's because the person that discovered his name was Theodore Escherick. So Escherichia, named after him, right? And then coli. Coli sounds like the colon, right? So Escherichia coli is actually found in the colon. It inhabits your large intestine. So that's like, oh, the name kind of tells you like what's going to be happening with that one. There's another microbe too that's called the Dilatidium coli, because it's found in the colon too. The names kind of tell you sometimes where they're going to be located at. Another thing the names can sometimes tell you is description. So like, what does it look like? So Staphylococcus aureus, the name tells you everything about it, what it looks like. So staphylo is actually a prefix that's going to tell you the arrangement of the bacteria. And so staphylo refers to cluster. It looks like some grapes, right? That looks like a cluster of grapes right there. And then the coccus is referred to as spherical, so circles, the grapes that I was mentioning. So they're gonna be grape-like in structure, gonna be clustered together. So whenever you look at staph on the microscope side, you're gonna know it's staph. You'll do other tests to like confirm, right? But you're gonna know, because it's kind of obvious with those guys. And then aureus. Okay, so when talking about aureus, y'all know the scientific symbol for gold?
AU.
AU. Do you guys know what the AU means? AU is Latin for aureum. The AU is aureum. So aureum is Latin for gold. So Staphylococcus aureus is gold. You're gonna see it in the lab too. Whenever we grow staph on some places, it's gonna be yellow. It's gonna be gold. Alright, because it grows gold. Alright, and so that's a key indicator between Staphylococcus aureus and Staphylococcus aureus. And I'll show you guys all that great stuff. Alright, so with the names, I know they're hard, but a lot of times if you actually look at it, dissect it, it tells you a lot about the bacteria. If you really just look at it and take some time. Alright, if you ever have questions about like some names and stuff, ask me. I like this stuff, right? It's fun. So ask me and I'll break it down. Alright, so one thing too that's really interesting is that after the first use of these scientific names, we want to abbreviate them after that, right? So we said the first time, we'll spell it all the way out. We'll say like Escherichia coli and Staphylococcus aureus are found in the human body. Cool, right? But then after that, when we reference it that second time and furthermore, we just write the first letter, period, and that genus or specific epithet follow along with that. So E. coli is found in the large intestine and Staph aureus is on the skin, or S. aureus is on the skin, right? Makes sense? That's only gonna be applicable when you guys start doing your lab report. It's not really useful to you like in the lecture because we don't really write a lot about them like that. On the exams you write stuff, but not that much like that, right? But on the lab reports, that's when it'll apply. Question, okay, so when I ask you guys questions, don't shout the answer right away because I know like me, I process fast, right? But everybody's not like me. You might process slower. You might process the slowest, and that's cool if you process slow, right? We're just gonna give you some time to process. So what I'll do is I'll read the question, I'll read the options, and we're gonna pause, and we're gonna let people think, and then we'll come back and we'll talk about the answers. Sounds good? Let's roll. So for the question, how would you write the scientific name name for humans on loose leaf paper? Is it A, Homo sapiens, like sideways, italicized, slanted, however you wanna say it? B, Homo sapiens underlined. C, Homo sapiens, just regular. And then D, Homo sapiens underlined regular. The B is Homo sapiens underlined, it's kinda slanted, you see that, like that's italicized, the B. It's italicized and underlined. So let's think for a second. Shouldn't think too long, so we kinda.
Oh,
Wait, you gonna tell me the answer? No, let's wait. But thank you for your attention to the one to get it done, I appreciate it. Not yet. Alrighty. Alright, so, feel like y'all ready, cause we kinda did it, on 3. 1, 2, 3, show me what you guys have. I can read sign language too. A, B, C. I can't. Yeah, there we go. Okay, we got D over there. We got some B. Wait, D. All D's? This is the only time we're gonna accept D's. Alright, good job. So, Homo sapiens, correct answer, letter D. Alright, types of microbes. Okay, so types of microorganisms. We're gonna talk about these guys, and the good news is that right now it's an overview. But we're gonna have specific chapters for bacteria and archaea together. So chapter 11 is bacteria and archaea together. Chapter 12 and 13, chapter 12 is fungi, protozoans, and algae. And that's what I write. Chapter 12 is fungi, protozoans, and algae. Chapter 13 is viruses. And then we have multicellular animal parasites as types of microorganisms as well. Alright, so we're gonna go over these guys, but first, I have a little task for you guys to do. I want you guys to organize these categories, or these groups, into their respective categories. So I want you to tell me where they go. Are they prokaryotes, or are they eukaryotes? And we'll talk about what that means in a second. So let's see, where would I put these guys? eukaryotes?
You go.
What's Joshua?
Yeah,
Yeah.
I'm trying to remember, but I'm pretty sure they're organisms with no compartmentalization. So like they don't have organelles and everything.
That's the word. Great job. Yeah. You wanna add anything to that,
Yeah, I
Josh?
think you one with single cell, one with multi-cell, but that's incorrect.
That's not always the case, but I
Alright.
understand why you said that though. Well, prokaryotes typically are
Like generally.
single cellular typically, prokaryotes are single cellular typically, but not always. And I know why you said it though. We'll talk about that. But they do not have organelles. They have something else too. That's one of the key nucleus. Yeah. So the name pro means before the carrier part right there is gonna be the nucleus. So these exist before there was a nucleus. They're older too. They're like 3 billion years old, whereas the 3 million years old, whereas your eukaryotes are 2.1 billion years old. So with your eukaryotes, EU means true. The EU prefix means true. The karyo, middle base word means nucleus, right? So eukaryotes have a true nucleus. Prokaryotes do not have a nucleus. Just like Joshua said, prokaryotes do not have compartmentalization. They do not have membrane-bound organelles. Eukaryotes do. Right? Eukaryotes do. So where are we gonna be putting our first one, bacteria? Where is it gonna go? Are you medical macro? Here, let's grab a packet off the table. Let's make sure it's one packet first. I took a page out. That's whole pack. Whole thing. It's Merry Christmas. You're welcome. Alright, so bacteria, where does it go? Prokaryotes. What about archaea? Where does that go? Prokaryotes. What about fungi? Where does that go? Alright, eukaryotes. What about protozoans? Where do those go? Okay, eukaryotes. Algae, where does that go? Viruses. Why? None, neither. They don't have cells. And because they don't have cells, are they living? No. So viruses are non-living because they lack cells. Viruses are acellular. Alright, so if somebody ever asks you, like, how do you know a virus isn't alive? Well, because viruses are acellular, A means without, they do not have cells. That's the main thing right there. Because viruses can look alive. That's the funny thing about viruses. They can trick you. They can look alive, but they're not. They're not. And I'm going to tell you some more details in just a moment. Alright? So viruses don't go in there at all. What about multicellular animal parasites? Eukaryotes, they're worms. Like, that's kind of what we're talking about, some worms there. But yeah, so that's going to be your eukaryotes. Some worms are really too small. Like, pinworms, can't really see those like talking about unless, of course, like, you gotta, at the egg stage, of course, you're not gonna see pinworm eggs. But, the worms themselves, you can wind them out of a womb and you can see it. Yeah. So certain stages of it, not gonna be useful. So here we have figure 1.2. Was that cool? Like to organize a little activity? Y'all like those little activities sprinkled through in there? Alright, we're gonna keep doing that then. So, question, what's your name?
Evie.
Abby?
Evie with the E.
Evie.
Yeah.
With a V.
Yeah.
What's that?
Oh, yeah. So for viruses, you said it goes on neither, right?
Nothing. Yeah, viruses are not living. They don't go anywhere. They're not alive. No. No cells, they're not alive. So, we're talking about figure 1.2. We have part A, we have some bacteria, those are Q. We have sporangia, which is gonna be important. We're looking at fungi. We have some amoebas right there, part C. We have some, that's algae. And then E, we have viruses. So I took the pictures and I put them next to the information that it should go to. So the first one's just like by itself. So let's look at the pictures with words. Alright, so those are the pictures by themselves. Now let's look at them with words. So for bacteria, that first picture is in fact bacteria, and we said that bacteria are going to be prokaryotes, right? So this is before the nucleus. So they don't have a nucleus because they existed before the nucleus was even a thing, right? And so these guys are single-celled organisms. They're unicellular. Uni means one cell. Okay, this is the thing. I will underline this for national next to this. We're gonna learn a lot about this in chapter 3. Peptidolycan, cell walls, peptido. What's that sound like? Peptido. Daniel? Yes, amino acids, alright? Why did you say that, amino acids? absolutely correct. So amino acids are connected together through peptide bonds. So that tells me something about some amino acids gonna be here, right? So peptidyl, something with amino acids. What about the glycan? What's that sound like? Sugars. There's something about amino acids and sugars, right? So we'll go into the details in chapter 3 just today. For chapter 1, bacteria, hapatidoglycan, and so on. The end. Alright. They also divide by binary fission. What's that? Divide means two. Fission means something's gonna be pulled apart, right? So basically, one cell is gonna divide into two cells. Exact replica too. It should be an exact copy. Alright? So binary fission is the way in which bacterial cells will divide. It should make an exact replica of itself. Now, another thing that's really cool about bacteria is that they get their nutrition from organic or inorganic chemicals or through photosynthesis, and we'll talk about that. So in chapter, 11? We'll talk about the ways in which the microbes get their nutrition. Alright, they can do it through organic chemicals, inorganic chemicals, or photosynthesis. With all those things, we'll be making some ATP, which we know is really important for energy, right? So chapter 5 is when we'll start talking about ATP production. And then bacteria, they can move. They can swim, right? Swim, move. And they do so through appendages called flagella. So flagella are like these long whip-like extensions that can be either like in one spot, they can be all over the body, just depends on what we're talking about. Alright, but nonetheless, they'll aid the movement of those bacteria. Alright, are we cool on bacteria? Can we go on to the next one? Alright, we're still talking about prokaryotes, but now we're gonna hit the archaea or archaea. So archaea, when I was in school, I was like, archaea, sounds like archaic. These are old bacteria, is what you're telling me? So these guys typically were discovered, they're referred to as being discovered first or earlier because of their characteristics and behaviors. So let's look at that, what does that mean? So, these guys do not have peptidoglycan in their cell walls. Alright, they don't have it. Alright, so make sure that's a key distinction between them and bacteria, they do not have it. They may not even have a cell wall altogether, it depends on what you're talking about. Alright? Now, when looking at these guys, they often live in extreme environments, right? So that kind of makes sense if you think about how they describe the early Earth, where you're devoid of oxygen, you have like those different gases such as like nitrogen, methane, you have your, what's that other one? CH4 is methane. Nitrogen gas, you have different anaerobic type gases there in that environment. Which makes sense to where it's like, okay, so here you have methanogens. Methanogens are microbes that live in methane-rich environments. Alright? So an example, have you guys ever been to a landfill before? Or passed by a landfill? I know you passed by one. You might not have been there, like why are you at the landfill? But you may have passed by a landfill before, right? So with a landfill, they typically have trash and they stack it. And so when you see that trash stacked at that lowest level, it's kind of gonna be devoid of oxygen once you have all that stuff like compressed and compacted together. And you can potentially get some methanogens growing there because of the fact that methane gas, there's no oxygen. So what's there? Anaerobic gases. So methane gas could be there, and you got those methanogens hanging out. Alright? Extreme halophiles. Halophiles, that means these guys, they love or have an affinity for salt. Right? So halophiles live in extremely salty environments. Have you guys ever seen the Great Salt Lake over there in Utah? Beautiful. I was actually driving, when I moved out here, I just moved I moved recently from Minnesota. From Louisiana, but I moved from Minnesota. Long story. But when I was driving, I drove through Utah and I saw the Great Salt Lake. I wasn't texting and driving. I was just driving. I had one hand on the wheel and I was like, just driving. I really was. I will not lie to you. I have some good videos too and some pictures. It's beautiful. You can see all the colors because halophiles, they typically have colors to them. You can see them. And you can see that the area was like extremely salty. If you guys ever go to, what's that other place called? The Yellowstone National Park, you'll see thermophiles there. You'll see thermophiles, extreme thermophiles in Yellowstone National Park. What do you think thermal means? Heat, right? Something hot. So that makes sense. At Yellowstone, you have those geysers that are really hot. You're not supposed to go by them, right? They're too hot. That will dissolve your skin. Like that can kill you. You don't go by that, right? So extreme thermophiles are beautiful, right? But they're extremely able to grow in hot environments, and you don't ever get by those, right? So just a couple of microbes to go out there. Now the thing about archaea is that we don't know right now of any that cause disease, right? Right now, I always throw right now out because it's been a lot of unprecedented times. So hopefully we don't cause diseases, but right now we don't. All right, fungi. picture B. And what's really cool, we're gonna talk about this in lab, and we're gonna actually look at this pharyngia, the little sac that hold all the little pollen grains and stuff, if you're talking about the pollen grains for plant things. But for this one, it's gonna be the different spores. Alright, so it's gonna be different spores for these guys. So we're talking about fungi, they are in fact eukaryotes. Alright, they're eukaryotes, that means they have a nucleus. They have membrane-bound organelles. The nucleus consists of the cell's genetic material, the DNA, deoxyribonucleic acid, we'll do that word later too. And it's gonna be surrounded by a nuclear membrane. We also have chitin cell walls. Have you guys heard that word before, chitin? You have, you just probably don't know. So chitin's a polysaccharide. Remember I was talking about cellulose? It's a polysaccharide. Poly means many. Saccharide means sugar. It's made up of a lot of sugars. Alright? So chitin, if you've ever stepped on a cockroach before, that little cracking sound you hear when you step on it, you're destroying the chitin. Alright? That's the body of the cockroach. A lot of insects are made of chitin, so we'll see that as well. So the same chitin that we see in fungi, fungal cell walls, the same chitin that we see in insect body planes. Which is pretty cool. And so fungi absorb organic chemicals for energy. Yeah, makes sense. We're talking about some processes called glycolysis. I don't know if you know about that yet, do you? No? Okay, we'll learn about that. But nonetheless, those cellular respirations, we're gonna be seeing that process of making energy. And we also have yeast are one type of fungi, then we have molds and mushrooms as another type of fungi. Alright, so yeast are unicellular, right? You guys have seen yeast before, baker's yeast. You eat bread. I love bread. Probably shouldn't eat as much as I do, but you know, it is what it is. And so when talking about, there's also yeast infections, we'll talk about that as well. Alright, but those are singular, circular, little, or oval, really not really circular, but oval in their appearance. Whereas the molds and mushrooms, I could see that. Right? I could see the molds and mushrooms. Like if you grow bread mold, you're gonna see the bread mold. Right? Mushrooms outside, I definitely see those, right? So these are multicellular and they typically are visible with the unaided eye. And so molds consist of masses of mycelia, we'll look at that are made with different filaments called hyphae. Alright, so we'll look at those great things a little bit later. The next one, protozoans. So protozoans are also eukaryotes, and these guys have the ability to absorb or ingest organic chemicals. Now one thing about prokaryotes is that a good bit of them sometimes are gonna be aquatic. Most of the time they do live in water. A good bit of the time they are aquatic, sometimes they're outside the water, but for the most part they are aquatic. Now, when talking about these guys, they can move, they may be able to move. And if they can move through pseudopods, and we call that process pseudopodia. So let's say for instance, this is an amoeba, and this amoeba is gonna be consuming this food particle, and they're gonna be using these extensions, the pseudopods, to like wrap around and grab the food particle. I'll show you a video in a second to see like what that looks like. And then you also have motility through cilia. So cilia look like, you guys ever seen like those pillows that people's houses would have the springs all around them? Little small structure, that's cilia, but within the body, right? Within the body or around the body of these protozoans, but they're short and they're gonna help the movement. And then the flagella is like long, the flagella is like the one that's like long with like undulating motion, like it's really big, right? It's a flagella. And so with protozoans, they can either be free living, they can hang out by themselves, they don't need a host. But if they are parasitic, they need a host. Right, so parasites need a host. Okay, so parasitic organisms, they need a host because that host has some type of nutrition or nutrient for it to thrive off of. Alright? And then some are photosynthetic. Yep, some are photosynthetic. And one thing that's really interesting about protozoans is that they can either reproduce sexually or asexually. So I put this video in Canvas for you guys. I'm not gonna play the whole thing because of, time, but it is pretty cool because I'm gonna just show you, point out to you guys what is going on for this particular one, because it is, I like it. No. Alright, so let's see. Alright, okay, alright, now skip the video, skip. Alright, okay. So, this is an amoeba. Which is a protozoan. This is Stentor, which is another protozoan. And it's pretty cool. Well, it's not a protozoan. Stentor is actually a ciliate, which is in the group of protists. Alright, we'll talk about that. But yeah, if you watch the video, you'll see like the little cilia, because this is a ciliate. It has the cilia, the little small whip-like. It's just going those little short appendages. And then the pseudopods, that's the whole body. Like all that stuff that's moving, that's like its little organelles or whatnot. But the pseudopodia, like its little feet that's gonna false feet, false pod feet. So they're gonna extend out and wrap around that Stentor ultimately to try to eat it. Alright, so it's pretty cool. I'll let you guys watch it. I like it. Do y'all wanna watch me speed it up? Wanna see what happens? I'm gonna speed it up. Alright, I'll speed it up, cause I do know how to speed it. I sped it up once before. Playback speed, let's speed this bad boy up. 1.25. Nah, go faster. 4. No, that's kinda crazy. You know what? 4s. Alright, let's do 2. Alright, so you see the amoebas wrapping around it. And it's kind of trying to gradually like consume that ciliate, so it's trying to shrink that body up. So it got all this is gone. It's like Merry Christmas is all mine. All that's mine now. The whole rest of the body is now the amoeba's nutritional, meal. So the stentor though is trying to get away. It's like, let me go. And so then you have the amoeba still holding on though. And this happens naturally in the environment, right? Because they have to eat, right? And it just so happens that despite it being bigger, it couldn't sustain this amoeba. Couldn't beat it. Or, not gonna say couldn't. All this is now the amoeba stuff. All that. So you see the amoeba's like, I'm trying to leave. Nope. Amoeba's like, I'm gonna come around. I'm coming. I'm gonna try to keep coming. And the stentor is like, no. And so the amoeba is probably really happy now to get all that food. It's probably really happy, like, yes, I got food, nutrition, great, but I want the rest of it. And this stentor is like, no. The amoeba's like, yes. And they're still trying to go. See how they're really trying to compete for this. They're trying to get it. The amoeba's trying to get it. But the stentor is like, I think I can get it. The little small ciliate right there, the cilia is helping it. Right, it extends out to try to regenerate that portion of its body. So ultimately, come on, you can do it. Right? And so you see how it got most of the body though, right? That stentor, most of it is already up in there, but by this being a ciliate, it's a protist, it has the ability to extend its body out. And it's now going to be real what it got. Yep. So when looking at it, the purpose of me showing this is for you guys to actually see what pseudopodia look like. Right? So the false feet, like the actual movement, it's not feet, but it looks like it's going to walk or move or whatnot. And then the cilia, little whip-like motion, right? And we'll look at the flagella later. But yeah, cool stuff. The video is in Canvas for you guys as well. If you go to the modules in week 1, I already put it there for you guys to like refer to if you like to. If it's weird to you, then don't go back to it. But if you want to watch it again, hey, go back to it. Alright, so protozoans. Okay, then we have algae. Alright, so we're looking at algae. Algae are going to also be eukaryotes. Algae are pretty cool because algae are very, very similar to plants. They have cellulose in their cell wall as well. Cellulose, like plants have cellulose. And so algae, funny enough, are grouped with the protists because they're similar to plants, but they're not really plants. Alright, they're similar to plants, but not really plants. So they're grouped with protists. And so these guys are found in fresh water, salt water, and even in the soil. And we know that they can go through photosynthesis, right? We know that algae is going to be very useful for making more of taking up more of the carbon dioxide to make more of that oxygen, right? Algae is really good for that. They produce oxygen as well as carbohydrates, food source. And then they can have reproduction as sexually reproducing organisms or asexually reproducing. This depends on what we're looking at. This is a picture of colonial algae, Walvix. That's not one that we look at in lab. I think we might, so I might order that one. Or ask to order that one. But yeah, it's a type of algae that has like small colonies of the bacterium in there. But like, I don't think I'm gonna order it because it's not infectious. It's just a regular old algae. You don't need to know that. Right? It's fun, but like, you need to know it now. Alright, so viruses. Viruses are acellular, right? So remember I was like, viruses are non-living because they don't have cells, right? Viruses can look like they're alive. They can mutate, we saw that. They can grow, we saw that, right? But they still need a host cell for survival. The only thing, they don't do metabolic functioning either. So it's like, yeah, you gotta be able to do that as a living organism. They can't do it, alright? So viruses are gonna have either DNA or RNA core, but never both, right? It won't have both DNA and RNA. It's gonna have either DNA or RNA. Also, viruses are gonna have a coat, right? They're gonna have a coat, a protein coat that's gonna enclose this nucleic acid. So viruses will always have nucleic acid and a protein coat. Alright? And the coat can have an envelope around it, a lipid envelope around it. That's a possibility, that's optional, right? So required DNA, RNA, so nucleic acid is required, protein coat, and then the envelope is optional. So these are replicated only when they are inside of a living whole cell. They are inert or non-living or non-functioning based. So inert would be non-functioning outside of a living host. This picture right here just shows us these little virions. Virion is referred to like an individual virus particle. So 3 virions, and you can see that there's some stuff around it, and then you can see like these little structures. And we'll talk about like what those little things are.
So the protein code, is that like a nucleus coronavirus?
It's like this. What's really interesting is that, let's say for instance I got influenza. So the protein code would be like a little structure and nucleic acid on the inside. My favorite one is bacteriophage. Alright, so bacteriophage is a type of virus that is going to infect bacteria. So bacteriophage is a virus that infects bacteria. The nucleic acid is on the inside of the protein coat. The protein coats can look different, it depends on what you're looking at. So like this is a icosahedral head type shape. This is like a little circle-like shape. You got some that can have like a, I can't draw it. I'm not gonna lie to you. I'll show you, cause I'm gonna mess it up. I'm not gonna. 3-dimensional sometimes doesn't work for me. I need my art person that makes the art movies and, yeah, cause I can't do it. Alright, types of viral protein coats. Alright, let's see. Here we go. Ooh, let's see. Let's see what we got. Oh, some research art. Alright, cool. Let's just go. Let's go. Come on. Here we go. There we go. Okay, so we're looking at these viruses, these different types, these shapes and stuff. So if you look at it, the little brown parts are gonna be the protein coat with the nucleic acid on the inside, the little squigglies of the nucleic acid. So it looks different depending upon what the virus is that you're talking about. So like this is that bacteriophage, a little funny weird head one I was talking about, the nucleic acid is on the inside. Same thing, nucleic acid on the inside. The little circle right here, nucleic acid is on the inside. So it always will be like surrounding nucleic acid. And if there's something around the protein coat, we'll know. Like coronavirus would look like, this. You'll have your nucleic acid, you'll have the virus in the protein coat, and then you'll have this little envelope on the outside. And then you got spikes too. Whole other thing. We'll talk about that too though. Alright, so DNA, rRNA, well, corona is RNA virus. So RNA, protein, envelope, spike. That works? Sure?
Yeah.
You're welcome. Alright, so, any other questions? Y'all wanna stretch? She said, please. Alright, so let's see if we hit this last one right quick and we'll stretch. Alright, so multicellular animal parasites, these are gonna be eukaryotes, multicellular organisms. They're not always strictly microorganisms. Alright, they can be visible by the unaided eye. So remember I mentioned the worms? I said that you have like pinworms, you have parasitic flatworms, roundworms. Roundworms are outside. Like I don't know, it should be outside here in Cali. Like I'm from Louisiana. If I go get a shovel back home in Louisiana, just go outside, start digging, I'm gonna hit some roundworms. We should hit some here too, 'cause it's very agricultural. So if I go outside into like the grass over there, get a shovel, I'm not gonna do it. But if I were to, I should be able to dig a little bit, roundworms galore. Because roundworms are highly abundant, naturally highly abundant. But with the worms, you can see worms. I can see worms, but you can't see them at the microscopic stage of like eggs. You can't see eggs. If we could, we wouldn't be having people getting infected with these types of infections by consuming the eggs. Right? You can't see eggs. So let's go ahead and stretch, and we're gonna come back in about like 2 minutes. Let's stretch right quick. A little 2-minute stretch.